Hi, I'm Emma Swanson. I'm going to tell you about DNA technology. DNA technology, it sounds pretty boring, right? Well, kind of is, but it's actually really important. Can help with so many birth defects and mutations and just shape everything about it. And there's a lot of controversy about it, but I'm just saying the facts with this. And you might be thinking, what is DNA technology? Well, as Khan Academy puts it, it is the sequencing, analyzing, and cutting and pasting of DNA. So in basic terms, that means you can move DNA throughout the sequence. You can move DNA throughout the sequence and move it to different spots, or you could full on remove portions of the DNA. And so there are different forms of this DNA technology. And so I'm going to tell you there is DNA sequencing, polymerase chain reaction, DNA cloning, and gel electrophoresis. So I'm going to go with a twofer for this one. I'm going to go explain DNA sequencing and gel electrophoresis. So DNA sequencing is like a laboratory technique used to determine the exact sequence of bases. And bases in short terms, as we would call them, A, C, G, and T. Or if you're going from RNA, it would be A to U. That's a different thing. Those are the bases in the DNA molecule. You have to go through a process called electrophoresis. And electrophoresis, DNA to be sequenced is placed at one end of a gel, which a gel is like a slab of gelatin like substance, but electrodes are placed at either end of the gel and electrical current is applied, causing the DNA molecules to move through the gel. Smaller molecules are going to move more rapidly, and so the DNA molecules will start to separate into different bands. Like there's the small ones, there's the big ones, like it's according to their size. And when you do that, that's how you're going to be able to separate and find out, oh, this is this, this is this. That's the short version of DNA sequencing. So let's talk about polymerase chain reaction. The polymerase chain reaction, that is used to make millions or billions of a certain DNA sequence. Like if you want to make a bad DNA sequence or a good one, you can make tons and tons of those. And in order to amplify a segment of DNA using PCR, that's it for short, the sample is first heated so the DNA denatures or separates into two pieces of a single strand of DNA. Next, an enzyme called TAC polymers synthesize, and it builds two new strands of DNA using the original strands as templates. The process results in the duplication of the original DNA, with each of the new molecules containing one old and one new strand of DNA. Then each of these strands can be created to make two copies and so on. Like 2, 10, 20, 100, thousands, leading to more than 1 billion exact copies of the original DNA sequence segment. And it seems like this would take forever, but it actually only takes a couple hours. Like, that's really cool to me. But that's how you are able to make a whole bunch of different copies. So let's go to DNA cloning. DNA cloning is, as it sounds, you're cloning DNA, and it's different than the PCR. When you clone your DNA in a typical cloning experiment, a target gene is inserted into a circular piece of DNA called a plasmid. The plasmid is introduced into bacteria via a process called transformation, and bacteria carrying the plasmid are selected using antibiotics. Bacteria with the correct plasmid are used to make plasmid DNA, or in some cases, induced to express the gene and make protein. Expressing the gene and making protein is way different than PCR, so it kind of sounds like PCR, but PCR uses a completely different process than DNA cloning. So we're able to express certain things in the gene. And that was like the last of DNA technology we're going to talk about. And I know that wasn't the most interesting topic to learn about. And I hope you were able to learn something new through it. And the good thing about this is, learning about it, is the fact that we're using it more and more. We're experimenting it with human genes more. We're going to try using it with humans. So I feel like it was a good thing to talk about. It can help with so many diseases and mutation, like the DNA cloning, the PCR, the sequencing, and the gel electrophoresis. DNA technology is a really helpful thing that we're going to be exploring more, so you might be hearing about it more in the future. So I want to say thank you so much for listening. I'm sorry it was such a boring topic, and have a nice day.